right, ladies and gentlemen, the books is in. The books are in. Yep. They in. They're here. ETM Hotel, Rennie, Shawn. Welcome in peace. My name is Shawn. I know everybody's chilling. Happy New Year to everybody that is actually in the chat right now. And we'll catch the replay of this particular video series, episode 20 of Science with Shawn. I hope y'all having a very productive day. I don't know if y'all had to work or whatever y'all got going on, man, but, you know, I hope all is well. Again, as you come in, you know, like the show, share the show. Let's see if we can get it cracking. And this thing, you know what I'm saying? It's the first live on the Mossy 2021. So let's have some fun with it, man. Let's see what we can kind of knowledge come out of this. You know what I'm saying? So just bear with me one moment. I'm actually sharing a link in a couple of groups. Appreciate everybody that's tuning in. I see Nunu, Juju, Nunu and Juju. Chavis. I see Rastaman in the building. I see you. Okay. Couple people coming in. All right. I know we got some onlookers that are not coming, so peace to y'all. I don't really expect a bunch of people, you know, because after, you know, last night for some people, they just not getting up. They might be hungover, still in the bed, or they might be watching Alabama practice on Notre Dame. You know what I'm saying? Because that's what I was doing. And then I was like, nah, enough of this. Just let me get to the national championship game. I already know who it's going to be. You know what I'm saying? But how y'all doing today? What up, LB? It's good, brother. Again, y'all, you know, help me like the show, share the show. And we're going to get it cracking. I see we got thousands of people up in this joker right now. Number one troll in the building. The number one trizzle. Yeah, man. I don't know if I'll be joined by anybody today, and it's all good. I ain't tripping. Y'all can hold it down. Yeah. A whopping 9,000. It was 11. You know, so people hop on and get nosy, then they get gone and be like, psh. You know what I'm saying? They want to see who it is or what's going on. Sad case. But yeah, I know a lot of people are watching that game and you know, they might not be like, forget what Shawn talking about. He ain't talking about nothing. Yeah. It's all good. But hopefully people will catch it in a replay and actually learn from this. So before we get going, man, let me check one more thing, and I'm gonna see if let's see something. I need to see something real quick, and then we'll get it crack-a-lacking. Man, I hope y'all ready for today, cause it ain't no secret what today is really about. You know what I'm saying? It ain't no secret. What up, Rostix Music? What's good, Black African Power? That's what I kind of figured. I kind of had this feeling. That was gonna be what I thought it was gonna be when I looked at that information right there. All right, so I see we got a few more people in here, man. You know what I'm saying? Like the show, share the show. Let's try to get this thing cracking, y'all. Let's get it cracking, and let's have some fun. All right. So as people coming in, man, they go probably catch us in the middle or something, but I'm gonna get to it so that you know, they can catch up or catch the replay, either one. Like I said, I ain't expecting a bunch of people today. They hung over. You know what I'm saying? Some headaches. I am though trying to see if I can get this dude with a whole lot of questions to hop in. That's gonna be the case for him. Let me see. Yeah. Happy New Year's, everybody. Yeah, New Year. Y'all must didn't hang out. Y'all stayed Rona free, huh? That's what's up. What's up, everybody that just not joining? Yeah, like the show, share the show. Let's get it cracking and see what's going on in these COVID streets. Can I get one more like and a share? All right, so I'm gonna share my screen and we're gonna get to the good, right? That's what we're here for. We're here to learn, so let's learn. All right. So got a little treat for y'all today. And hopefully, you know what I'm saying, y'all appreciate the treat, receive it well, and let's see what we can uncover and learn. So what did COVID-19 teach us? All right, now we know a year ago, um, they began to identify SARS-CoV-2, coronavirus, or COVID-19, right? They began to identify this. Now, a year later, we've come to know more about SARS-CoV-2 and especially COVID-19, right? So I just want to give you a little recap and then we will move forward. So here's a quick outline of today's actual lessons. All right, we're going to do a quick review of what SARS-CoV-2 or COVID-19 is. We'll have a couple of questions, like a quiz, you know, pretty much. I'll conclude, and then we'll have like a small Q&A. Then, you know, we'll get into our structure of viruses by Professor Vince Racaniello. And, you know, I put this in here and I didn't have to, but any material used during this presentation will be available in the description of the video. Remember, I always source up. So the sources will be in the description of the video. If I pulled from those sources, who knows if I did or did not? You know what I'm saying? So, Notre Dame to switch their quarterback. Here we go.
SARS-CoV-2, the virus causing COVID-19, is a coronavirus. Many different coronaviruses are known to infect humans. They typically cause respiratory infections of varying degrees of severity. In some cases, different types of coronaviruses may lead to gastrointestinal infections. In the last 20 years, coronaviruses were the cause of 2 outbreaks, SARS-CoV in 2002-2003 and MERS-CoV in 2012. In 2019, another coronavirus, SARS-CoV-2, emerged in Wuhan, China. To develop drug treatments and vaccines against SARS-CoV-2, it is essential to understand the structure and characteristics of this novel virus. Let's take a closer look at the structure of SARS-CoV-2. The genome of coronaviruses is encoded by a single-stranded RNA. The RNA is surrounded by a lipid envelope, which contains a number of different proteins. This gives the coronavirus the appearance of a crown. Crown in Latin is corona, hence the name coronavirus. So how does the virus infect human cells? The virus uses a receptor known as ACE2 in order to gain entry into a host cell. ACE2 receptors are highly expressed in cells of the nose, lower respiratory system, heart, kidney, gastrointestinal system, and endothelial cells in humans. Proteins present on the surface of the virus known as S protein or spike proteins bind to the ACE2 receptor, allowing the virus to attach and release its RNA into the cell. Once the viral genome enters the cell, SARS-CoV-2 replicates rapidly, But how does it do this? The first step in the replication process is for the host ribosome to bind to the viral RNA, and through the process of translation, produce a protein called RNA-dependent RNA polymerase, or RDRP. RDRP is an enzyme that can produce new RNA molecules by using an existing viral RNA strand as a template. So once produced, the RDRP can create new copies of the viral genome. But the RDRP doesn't always replicate the entire viral genome. Sometimes it stops early and creates shorter RNA strands, which are known as subgenomic RNAs. The host ribosomes translate these pieces of subgenomic RNAs into proteins. Specific enzymes known as proteases then come along and cut the proteins into individual structural proteins. Then the replicated viral RNA and proteins can be packaged up into a new virus, which is released from the cell and ready to infect another cell or another person. Based on what we know about SARS-CoV-2 and other similar viruses, such as SARS-CoV, researchers have identified several potential treatment targets. We've seen how important RNA replication and protein synthesis are for viral replication. So it's not surprising that many potential treatments target these processes. Remdesivir, a broad-spectrum antiviral drug, inhibits the RNA-dependent RNA polymerase and may block viral genome replication. Lopinavir, a drug used in combination with ritonavir to treat HIV, binds to specific SARS-CoV-2 proteases, preventing the proper translation of viral proteins. Blocking this pathway may disrupt the survival of the virus. Of course, there are many other possible targets for therapeutics. Blocking the interaction between the spike proteins and the ACE2 receptor wouldn't kill the virus, but it would prevent it from infecting a cell, and without a host, the virus couldn't replicate, which would severely limit its ability to infect. Although we still have a lot to learn about SARS-CoV-2, understanding its basic structure and how it infects host cells is the first step to identifying potential treatment targets against SARS-CoV-2. That's it for now. If you want to improve your understanding of COVID-19, make sure to register for a free
I ain't doing all that, bro.
straight trial account and check out.
All right, hold on. So now that we just did our little, you know what I'm saying, quick review, I got a couple questions for the people. Put your hands in the chat for me. So question number 1 is, what is SARS-CoV-2? A, a name of a car. B, medicine. C, a disease. D, a virus. Hey, don't be trying to use Google, man. Come on, y'all. We've been at it heavy this month. And all year, we should know what SARS-CoV-2 is. I'm say, all right. So I'm waiting, because if I split down the number of viewers versus how many answers we should have in the chat, you know what I'm saying? It's gonna take 9. It's gonna take 9 to rule. Y'all a little slow today, man. What's up? Y'all hungover? Y'all hungover? We're looking for 4 more answers. Where you at, Juju? Where you at, Keish? Where you at, number 1 troll? Somebody else must be live, you like. Did I interrupt somebody live? So the question is, what is SARS-CoV-2? That fool said she hungover.
They're drunk on pseudoisms.
Drunk off pseudoisms, bruh. Straight up. We need 9 to move on. 1, 2, 3, 4, 5, 6, 7, 8. We need one more answer. We got 18. Actually, we need 10. We got 18 now. We need two more answers. Can we get 2 more answers? I mean, I know a lot of y'all just listening in, but you know what I'm saying? It ain't nothing to hit the button. Just drop a little answer in there. If you don't know and you don't want to answer, you know what I'm saying? I mean, if you don't know, you know, just put an answer in there. Just pick one you choose. We all trying to learn right now. All right, we got a couple of more. Everything, yo, listen, I can't take that. I can't take that. Can't. All right, so we got a couple of more answers. Everybody got that right. Is D. What's the name of the disease? Anybody know the name of the disease? If SARS-CoV-2 is the virus, what's the name of the disease? There we go. Oh, Zane just got here. He late. All right, so here we go. Let's move on. Question number 2. Was SARS-CoV-2 the reason for the outbreak? Yes or no? Was SARS-CoV-2 the reason for the outbreak? Yes or no? All right, we need like 10. We need 10 answers on this. We got 2 yeses and 2 nos. We got 3 yeses and 2 nos. It ain't no trick question. It ain't a trick question. Was SARS-CoV-2 the reason for the outbreak? Keyword is outbreak. We got 3, 4 yeses. 1, 2, 3, 4, 5, 6. 6 no's. 5 yeses. Ooh, split. Cause we know, you know what I'm saying, we got more viewers, but a couple of them are not answering. So was SARS-CoV-2 the reason for the outbreak? Think about what I'm. All right, let me say it like this. Think about what I'm saying. If SARS-CoV-2, if we've never known SARS-CoV-2, Would it be responsible for what's going on? Is SARS-CoV-2 responsible for what's going on right now? Is SARS-CoV-2 responsible for what's going on right now? Y'all thought Chavis was gonna ride with y'all on this? So Tremis is saying no. Rastic says no. Lawski says no. Nunu says no. Rasta says yes. Rastic says evolution. People eating strange foods is what caused the Chavis, what's the answer, bro? Mika will get into that. I think we might. I don't know. We might get into that. We can get into that during the Q&A. Y'all was wrong like a mug. Chavis was right. Think about it. If we don't have the emergence of SARS-CoV-2, then we don't have an outbreak. It's that simple. Number 3. What did independent investigators uncover when investigating the cause of the spread of SARS-CoV-2? What did independent investigators uncover when investigating the cause of the spread of SARS-CoV-2? A, that the virus escaped the lab and it was developed by humans creating a bioweapon. B, the virus developed in bats and was accidentally discovered. C, the virus did not escape a lab. The virus is likely to have passed from bats to humans, probably through an intermediate animal. D, that the coronavirus is a Chinese virus. I got you, Trump. We need 17 ounces. I mean, I know we have a bunch of people that just joined the discussion. I prefer you guys to actually, you know what I'm saying, answer the question too. This is an interactive, an interactive quiz. It's not heavy. Even if you're not familiar, you know what I'm saying, we recommend it. But we need 16 ounces. And right now we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. We need about 6 more ounces. My bad. Now we need about a few more ounces. A couple people dipped out on us. Dang, I guess they didn't want to get into the bill. All right, a couple more people dipped out. Man, I'm running people off. What they want me to do is call some drama. Oh, I'm gonna call some drama. You best believe the drama coming. Drama coming. Joe Schmo, peace to you, brother. Never seen you in the chat before, but I appreciate you joining the conversation. All right, so it looked like we gaining a majority. All right, yeah, it looked like we got a majority. Yep, y'all on it. All right, we on it now. We back at it. Question 4. What does EUA mean? Does EUA mean Easy Umbrella Assembly, Emergency Unit Assistance, Emergency Use Authorization, Easy Unique Apples? So A is Easy Umbrella Assembly, B, Emergency Unit Assistance, C, Emergency Use Authorization, D, Easy Unique Apples. The question again is, what does EUA mean? Yeah, I sound like Rick Ross. What does EUA mean, ladies and gents? We're looking for 13. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. We need a couple of more answers. The question is, what does EUA mean? Easy Umbrella Assembly, B, Emergency Unit Assistance, C, Emergency Use Authorization, D, Easy Unique Apples. Come on, ladies and gents. Give me some answers. Couple of more answers. One more answer, be straight. Can we get one more answer from? All right, there we go. What up, Teddy Peace? I see you joining the conversation. Just not getting in the building. What's good? All right, so here we go. Question number 5. Do clinical trial results show whether vaccines are effective? Yes or no? A is yes, B is no. The question again, do clinical trial results show whether vaccines are effective? A for yes, B for no. Lowski, we're talking about clinical trials. You know how you go through clinical trials in order to get, you know what I'm saying, your EAU? You can't get the EAU without going through clinical trials. So do clinical trial results show whether vaccines are effective? Yes or no? Now I answered it already, but the majority had already put in. Now, Chavis, you sure? How do you get the efficacy rate? Chavis wrong on this one. Clinical trials, trial results show whether vaccines are effective. Yes, they do. He messing with y'all right now. But they do show the efficacy level. because that's how you get the, you know, from the data, that's how you get, you know, the level of efficacy. What up, Teddy? But yeah, Chavis messing with y'all. He trying to curve y'all. Yeah, we not being too technical right now. We just keeping it simple though, Chavis. And it does tell us if it's effective and infectious. If we want to be technical, the answer still be yes on both parts. Because it's effectively working right now in the community. We got 2 million people who's been administered at least shot number 1. And then, you know what I'm saying? The efficacy rate, of course, we all know is high because of the results. So yeah. All right. But just keeping it simple though. All right. Now, question number 6. Does the new vaccine alter or change your DNA? A, yes. B, I don't know why I got not in here, means no. C, potentially. Does the new vaccine alter or change your DNA? I'm gonna repeat it one more time. Does the new vaccine alter or change your DNA? A is yes, B is no, C is potentially. One, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. All right, let's move on. Bonus question. Are mRNA vaccines effective? Now, when I say effective, you can also interchange that word and put show efficacy. Are they effective? I'm gonna leave it like that. Are mRNA vaccines effective? A, not enough evidence to determine the answer. B, yes, they have proven to be effective. C, the jury is still out on this vaccine. Mika don't know, she guessing. He said B or C. All right, Chavis, I'm gonna get it to you like this. So, what you think gonna happen in April when Moderna and Pfizer go up for full authorization? You think they gonna get it, or you based on the data that you have now and your understanding of the vaccine, or do you think that they will be denied? Mika, every vaccine is always ongoing. Man, right. She's just gone. I'm waiting to see if Chavis will respond to my question. Do you think Moderna will get full approval in late April, or do you think Moderna and Pfizer will receive full approval in April? Because listen to what I'm saying. Oh, okay, all right. So here's what I'm saying, everybody that don't know what me and Chavis is actually talking about. Full approval would mean that after the data rolls in and they see just how effective this vaccine is against the coronavirus, they can opt in for the approval. Now, when it comes time for that full approval, that's when people like me and you, the layman, me, you, and everybody else, and all the people talking about they ain't getting the vaccine, that's when we'll be in line to get the vaccine. We not in line right now. Even though it's in EAU status right now, this thing will be in full approval by the time we'll be able to get it. And then they got to have enough doses to go to all the pharmacies. All right. Double bonus question. Why do experts continue to assess vaccine effectiveness in real world conditions? A, the reason for conducting additional vaccine effectiveness assessments is to make sure a vaccine protects people from getting the disease under real world conditions outside of the strict settings of clinical trials. B, there are many factors that can affect the vaccine's effectiveness in real world situations. These factors can include things such as how a vaccine is transported and stored, or even how patients are vaccinated. C, vaccine effectiveness can also be affected by differences in the underlying medical conditions of people vaccinated in the real world compared to those in the clinical trials. Vaccine effectiveness assessments can also provide important information about how well a vaccine is working in groups of people not included or not well represented in clinical trials. Or D, all of the above. Anybody need me to repeat that? We need about 13 responses. So again, the double bonus question is, why do experts continue to assess vaccine effectiveness in a real world conditions? A, the reason for conducting additional vaccine effectiveness assessments is to make sure a vaccine protects people from getting a disease under real world conditions outside of the strict settings of clinical trials. B, there are many factors that can affect a vaccine's effectiveness in a real world situation. These factors can include things such as how a vaccine is transported and stored, or even how patients are vaccinated. C, vaccine effectiveness can also be affected by differences in underlying medical conditions. People vaccinated in the real world compared to those in the clinical trials. Vaccine effectiveness assessments can also provide important information about how well a vaccine is working in groups of people not included or not well represented in clinical trials. Or D, all of the above. Few more answers. Alkaline Institute. Anybody else? Anybody else want to answer? Tremors, you didn't answer. You must have left. You don't understand the question? All right, one more time. Why do experts continue to assess vaccine effectiveness in real world conditions? Is it A, the reason for conducting additional vaccine effectiveness assessments is to make sure a vaccine protects people from getting a disease under real world conditions outside of the strict settings of clinical trials? B, there are many factors that can affect a vaccine's effectiveness in real world situations. These factors can include things such as how a vaccine is transported and stored, or even how patients are vaccinated. C, vaccine effectiveness can also be affected by differences in underlying medical conditions of people vaccinated in the real world compared to those in the clinical trials. Vaccine effectiveness assessments can also provide important information about how well a vaccine is working in groups of people not included or not well represented in clinical trials. Or D, all of the above. One, two, three, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. All right. Conclusion. SARS-CoV-2, COVID-19 has taught us a lot about ourselves and the environment we live in. This coronavirus has proven that nature is in fact in control, and at any time it can and will outlive humanity. We learned the meaning of social distancing, the importance of wearing a mask, and how viruses spread so quickly. This virus has shown just how important our immune systems are, and that herbs and foods cannot create an immune response against the virus. But we are still learning a lot from COVID-19, and we must not take this opportunity for granted. Appreciate everybody tuning in for that. Let's move on to our next phase. Anybody got any questions or answers about anything that we actually just went through? If you do, this is the time for you to, you know what I'm saying, type it in the chat and let's build on that. If not, then we're going to transition into the other half of this presentation. There's a lot that we did not go over or talk about, right? There's a lot. It could have been a lot more that we actually could have dealt with regarding, you know, questions and answers, questions and quiz questions and things of that nature. But I just wanted to kind of keep it simple and not overtly, you know, make people lose their minds about these things. So I don't see any questions or answers at this particular time, but if there are any in the chat or in the comment section of the video, then we'll address them accordingly. So again, you know, I appreciate everybody, you know, participating in the quiz. For those who was just looking and not answering, hopefully, you know, in your head, you was actually participating. I can't determine that, however, you know what I'm saying? But I do appreciate the view.
But before we do that, I want to talk about how viruses are built, because this really helps you to understand how they're taken apart in the next lecture. Now, first of all, the virus particle, when you look at it, it's made up of proteins, in some cases membranes, as we'll see today. They have very specific functions. Of course, they look beautiful on top of everything, but they also have functions. They have to protect the viral genome, right? That's one of their main functions. They have to be able to come together and make a stable shell. They have to recognize the viral nucleic acid specifically. Don't want to put cellular nucleic acids in the particle. That would be a waste. And there are very specific mechanisms for that, which we'll talk about later. And then for viruses that have an envelope or a membrane, I use the term interchangeably. The proteins have to associate with it. So here on the top are 3 examples of virus particles. On the left, influenza virus, which is enveloped. In the middle is an adenovirus, which is made of pure protein and nucleic acid, no envelope around it. And on the right, a bacteriophage, very different configurations. But my goal today is to really simplify all of this for you. So protection of the genome is really important. As virus particles go from host to host, they have to protect it so it's not inactivated. Another function is to deliver the genome once the particles inside of a cell. And so the viral proteins have to bind cell receptors. They have to uncoat the genome. That's a term I use for releasing the genome from the virus particle. I often call it uncoating. In many viruses, the ones with membranes, the membranes fuse with host cell membranes to release the nucleic acid. We're going to talk about all this next time. And then they have to bring the nucleic acid to the right site in the cell. It differs according to the nucleic acid. Sometimes the right site is the cytosol, sometimes it's the nucleus, and the viral proteins have a big role to play in getting the particle to the right place. Here are 3 more viruses. On the left is a rabies or rhabdovirus, an enveloped particle. In the middle, a toga virus, also enveloped. And on the right, a pox virus, much bigger. Before we start, let's do some definitions so you understand what I am saying. The first at the top there, subunit. When I say a subunit of a virus particle, I mean a single folded polypeptide chain. And so there on the upper left are some examples of viral proteins, VPs. These are often the abbreviations for capsid proteins, virus particle proteins. And those are each an individual polypeptide folded up. That's a subunit. Next, we have a structural unit, which I will often call a protomer or an asymmetric unit. This is the basic unit that we use to build up capsids. So here on the right is a capsid. It's a virus particle, and it is made of subunits. You can see the red subunit there, VP3, is part of the capsid right there, but it's also part of a structural unit. Here, it's made up of 3 of these proteins, VP1, 2, and 3, blue, red, and green. Okay, so subunit, structural unit, and then finally capsid is the entire assembly. Capsid is Latin from box, it's the protein shell that encodes the genome. So this is a word I will use a lot, capsid, and a version of it which I'll tell you about in a moment. Now for some viruses, there's an extra part of them, that's the envelope or the viral membrane. It's always derived from the host cell, because viruses cannot make lipids. So all viral membranes come from the host cell. So here on the left, an example influenza virus, that tan circle around the outer periphery, that's the membrane of that. And right is a herpes virus. And again, the membrane is the tan layer on the outside. And you can see these membranes are studded with proteins. Those are viral glycoproteins, which we'll talk about today as well. And finally, there's another term which is every year confusing. Every year I try harder. Nucleocapsid or core. The term can be used interchangeably. It is the nucleic acid protein assembly within the particle. We use the word nucleocapsid when it's a discrete substructure. So here in this influenza virus, the RNA plus protein, which are those little pieces inside there, there's RNA plus protein. Those are called the nucleocapsids. In this herpes virus on the right, you actually have a shell, a capsid. So the capsid above right there, that is in a herpes virus. It's encased in a membrane, and it has the DNA in it. And there we call it a nucleocapsid because it's a substructure. It has a membrane around it. The top right would just be a capsid, not a nucleocapsid. Well, the nucleocapsid contains the genome, and it protects it till it gets, in this case, to the nucleus. It's an extra layer of protection. Now, you may say that sounds arbitrary, and much of nomenclature is arbitrary, right? But this stuff is in the literature, so I want to make sure you know it. So that's a capsid, and here it's a nucleocapsid. If this herpes didn't have an envelope or a membrane, if that was herpes virus, it would be a capsid, not a nucleocapsid. Okay, so those are the terms I am going to use. All right, some size here to put things in perspective, some of the numbers I'm going to be using. Nanometer, I'll talk about nanometers a lot, 10 to the minus 9 meters, which is 10 angstroms or 0.001 microns, right? Those are all the same thing. We use this to talk about viruses and subviral particles because they're very small. So the alpha helix in a protein, here's an alpha helix on the top right, that's in a protein. That's about a nanometer in diameter. If you think about any typical protein. DNA on the middle there, is about 2 nanometers in diameter, about twice the width of an alpha helix. There's a ribosome, about 20 nanometers in diameter. Poliovirus, 30 nanometers, slightly larger than a ribosome. And one of the bigger viruses, Pandora virus, here at the lower right, 1,000 nanometers. So this is huge, and there's some that are slightly bigger. Pithovirus is slightly bigger than that, 1,200 nanometers. One concept that's very important for you to understand is that virus particles are metastable. What that means is sometimes they're stable and sometimes they're not. We call them metastable. So as I said earlier, capsid proteins, viral structural proteins, have to protect the genome as it travels around from cell to cell, host to host. So they have to be very stable. For example, respiratory virus particles have to be stable in aerosols that we produce when we breathe long enough so that they can reach another host and infect them. Some viruses infect the gastrointestinal tract. They actually pass through the stomach and the small intestine, and they remain stable, which is remarkable. Given all the conditions, they have to remain stable there. But at some point in reproductive cycle, when that virus gets to the right cell, it's got to give up the genome. It has to become unstable. So that's why we call them metastable, because sometimes they're stable and sometimes they're not. So here's an example of a virus that is outside of the cell, extremely stable. But in this case, when it binds a cell surface receptor, that's a trigger for it to give up its nucleic acid. And these sorts of activities we'll talk about next time. But the concept of metastability is inherent in the structure of the particle. Somehow the particle has to be built so that it lasts really well in the environment, but can come apart when it's on the right cell. Now, metastability is a consequence of the fact that virus particles, or virions, so the virion is a name that I use for an infectious virus particle. So when I say virus particle, it may or may not be infectious, but virion means infectious. Virus particles have not attained their minimum free energy conformation. So as a particle exists, it has to surmount an energy barrier to get to that minimum free energy conformation. So here's a illustrative graph where we're looking at free energy on the y-axis and with time on the right, if you will. When virus particles are built, energy is built into the particle, into the bonds of the particle. And we call viruses spring-loaded because that energy is then used later to disassemble the particle if the cell provides the right signal. If the cell provides a receptor or low pH or protease, number of different conditions. The energy put into the particle during assembly is used to encode it. And so to disassemble, to get from the stable to the unstable state, viruses have to surmount an energy barrier as shown here. So one is the infectious particle in the environment. To give up its genome, which would be state number 3, it has to get over a high energy barrier. And that's important because if not for that barrier, viruses would spontaneously be giving up their genomes, and that wouldn't be good. And to surmount that barrier, the energy built into the assembly of the capsid has to be liberated. And that trigger for that, as I said, is receptors or low pH or other conditions. So that's metastability. Viruses are metastable. How do you do this? How do you make a particle stable yet unstable? So first of all, stability is made by symmetrical arrangement of identical proteins. You're going to see in a few moments that many virus particles are built by taking one or a few proteins and repeating them multiple times. They have maximal interaction with each other, and that gives the particle stability. Well, how can it be unstable at the same time? Well, first of all, these proteins are not covalently joined. In most cases, they are non-covalently joined. And so they can be taken apart or loosened upon infection that provides the right trigger. So in the absence of the trigger, the virus remains stable because of all these interactions of many identical proteins. And under the right condition, the stability is reversed. So that's how metastability is achieved. So viral capsids are metastable because they must protect.
Nope. Nope. No. I seen y'all trying to cheat. Now I'm gonna let it play. I'm not gonna interrupt the actual question there, so I hope y'all following along. So, you know, he asked the question, and I'm gonna get it back up on the screen because my I actually clicked the wrong button when I got there. Yeah, so y'all got all of, dang it. There you go. There go the answer. Viral capsules are metatastable because they must protect the viral genome when the particle is outside of the shell. They must come apart and release the genome into the cell. Blah, 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 blah. They have not obtained a minimum free energy conformation. They are all spring loaded, or E, all of the above. That's a good question I asked the lady too.
Capsids are metastable because they must protect the genome outside of the cell. They must come apart and release a genome into a cell. They have not obtained a minimum free energy conformation. They are spring-loaded, all of the above. All right, how did we do here? All right, most of you got all of the above, which is correct. Every one of those things explains why viral capsids are metastable. They have to protect the genome. They have to come apart, both ends of metastability. They haven't maintained a minimum free energy conformation. They have to surmount a barrier to get to it, and we call that spring-loaded. I'm going to talk to you now about how viruses are built. And throughout this course, you're going to see really nice pictures of virus particles. And I just want to briefly tell you how we get those pictures. And that's the tools of viral structural biology. It includes electron microscopy, X-ray crystallography, cryo-electron microscopy, and cryo-electron tomography, and NMR. This is well described in the book in chapter 4 if you're interested in getting into it in some detail. The technology has improved markedly since the 1930s when the first EM was built. This is a modern day cryo-electron microscope that I visited at Cornell up in Ithaca. And that's the person who runs it. Look at this thing. It's huge. You have to have liquid nitrogen around to cool things. Lots of wires going everywhere. I don't know how people sort this out, but you can use this to solve structures. So 1939, 1940, of course, mentioned this earlier. Helmuth Ruska used an EM to see bacteriophages for the first time. Very important discovery showing that viruses were not liquids, they were particulate. And he published a paper here. It's in German if you want to go look it up. I see bacteriophagen lice. I can get that part. And that was important, but the tech has improved incredibly since these days. This image is taken by negative staining. Biological materials don't have much contrast on their own, so typically we have to stain them. And you know that if you want to look at cells in some way, you stain them with various types. You want to look at tissues, you stain them. It's the same with viruses. They don't have contrast, but you can't use the typical stains for viruses. You have to use an electron-dense material because remember, electron microscope, you're bombarding the sample with electrons much smaller than light particles, and therefore the resolution is greater. But typical dyes that you use with light microscopy, you cannot use with electron microscopy. So you use something like uranyl acetate or phosphotungstate. We call this electron dense. They scatter electrons. So you're basically coating the particle with the stain, and the electrons are bouncing off of it. So where the virus is no light is going through. It's a negative stain. The resolution is not great on this. It's about 50 to 75 angstroms. And as I told you earlier, right, the alpha helix is about 10 angstroms or 10 angstroms in diameter, 1 nanometer. So you can't really tell any detailed structural interpretation from negative stained EM. But in the early days, pictures like this were taken and gave us a lot of information. There's an adenovirus where for the first time they saw these fibers sticking out of the particle, and it was quite interesting. You can see there's something repeating about the particle structure. Here's a hepatitis B virus, an influenza virus, and a polio virus. So you get some information, but not enough detail to know how they are composed. Yet, just last week, they were taking negative stain electron micrographs of the new coronavirus to see what it looked like. X-ray crystallography now gives you a much, much higher resolution. You can get 2 to 3 angstroms resolution, and just maybe 40 years ago, this became possible with viruses. Initially, it was done with proteins. Remember, myoglobin was one of the first proteins to be crystallized. But then it was possible with viruses only because computers were developed to take care of the massive data sets that were generated. You couldn't deal with the data sets of a virus structure before computers. The way it works is, in the old days anyway, you had to get your virus to form a crystal. So you would purify tons and tons of virus and figure out conditions for it to crystallize. And that often took a long time, and sometimes it didn't work. Nowadays, you can buy crystallization kits, which are trays with different chemicals in each one, and you put your virus and see what conditions give you a crystal. Once you get crystals, you bombard them with X-rays, and you hope that the crystal diffracts the X-rays. Sometimes they don't, sometimes they fall apart, but in some cases they will diffract the X-rays. And in the old days, you used a piece of film to capture the diffraction pattern. If you remember the famous X-ray film that Rosalind Franklin took and Watson and Crick stole from her desk, they held it up, and that was a diffraction pattern like this. Nowadays, we can use detectors where you don't need film anymore. They're image detectors directly. But what this pattern means is where all the atoms are located. The X-rays are bouncing off the atoms in the crystal, and you can tell where each atom is based on their location there. Cryo-electron microscopy came around years later, and only 2017, the Nobel Prize in Chemistry was awarded to 3 individuals for their work in developing the technology to do cryo-EM. Jacques Dubouchet, Joachim Frank, who's up at the Medical School of Columbia, and Richard Henderson. And the way this works is you don't have to make a crystal. You take your preparation of virus particle and you freeze it very quickly in water, and that's enough to give it contrast. And that top is an electron micrograph of the frozen virus particles. And then you take images of many, many particles, hundreds of particles. And the idea is that each particle is oriented in a slightly different position in 3 dimensions. You take all of your pictures and you process them to make a 3-dimensional image. You do Fourier transforms, and you orient all these phases, and then you do a reverse transform, and eventually you can get your 3D reconstruction. And this now has gotten to very high resolution. It's approaching X-ray crystallography in terms of resolution, because not only is the tech being improved, but the computational approaches are really improving as well. Now, the cryo-electron tomography is a variant of this, where you don't have to photograph so many particles, but you take a particle and you tilt the stage, so you make your own 3-dimensional image of it. And a lot of the images that I will show you today were made by cryo-EM. So here's an example of the 2 technologies. This is poliovirus, whose structure was solved by X-ray crystallography in 1985. That was the first animal virus to have its structure solved, 2.9 angstroms, and you can see where every atom is in the 3-dimensional structure. Thousands and thousands of atoms in that particle, and you can get a coordinate list, XYZ coordinate list for those atoms, and draw pictures like this one, and you can color them as you will. This one is depth cued, which means the atoms that are farthest away from the center are light, and the ones closer are dark. So you can get an impression of the surface of the particle. On the right is the same virus solved by cryo-EM 20 angstroms. You can see you don't see any detail. But now, of course, you can do it at 2.9 angstroms, most likely. This is just to show you that at one time, it wasn't as comparable to X-ray crystallography, but cryo-EM you can do in a few weeks. And when new viruses are isolated, they can be done rapidly because you don't need to make crystals. So when Zika virus emerged in 2015, within 6 months, the structure was solved by cryo-EM. These are some images I made on our computer. You can download the coordinates in their programs, free programs out there that you can use to manipulate them. On the upper left is a space filling view. I've colored the proteins differently so you can see their arrangement. And here on the right is a ribbon diagram where I've zoomed in so you can see how the proteins interact with each other. So lots of very, very useful information you can get from this. This is one of the biggest viruses, Cafeteria roenbergensis virus. This was done by Shawn at the University of Texas, El Paso. This was only just published. It took him years and years to solve this. But he told me just to do like one turn of this virus requires 3 million CPU hours. There's so many atoms in this particle. It's over 300 nanometers, 15,000 capsid proteins. And he made this movie of it rotating on the ocean because that's where its host lives, in the ocean. This virus infects the host in the ocean. It's huge. He said it would crash his computer continuously while doing this, but they are able to solve the structure. So you can do a lot with the technology. Now, the other day I told you about the Baltimore scheme, 7 different genome types, right? Well, I can use the subway again to illustrate the different types of virus particles. There are only 3. You can put everything in this course in terms of 2 subway lines here in New York City. The number 7 genomes, the number 3 virus particles. There are 3 kinds of virus particles. There are particles with helical symmetry. And that's tobacco mosaic virus on the upper right, the first virus discovered. We'll talk about how that's built. There are viruses with icosahedral symmetry. Second down there, like poliovirus. And then the rest that don't fit into these 2 categories, we call complex because we don't really understand how they are built. They don't fit into any one of these 2 categories. They include pox viruses and pithoviruses and Pandora viruses. They're very, very big viruses with really no symmetry to them. And I'm going to tell you today mainly about how to build helical and icosahedral viruses. And this actually this story starts with Watson and Crick, who all of you know solved the structure of DNA in 1953, which happens to be the year I was born. And I always say that's one of the reasons I became a scientist, because somehow I felt that. But after they finished with DNA structure, they turned to virus particles. And they noticed that when you look at all the EMs of virus particles that were published at that point in the 50s, most of them were either spherical or rod-shaped. So here we have rod-shaped tobacco mosaic virus, and then there are other viruses that were subsequently discovered that were rod-shaped. And here, poliovirus is spherical. And so they thought, okay, viral genomes are small. Of course, that's what we knew back then. We know now they're big, but the principle is still similar. Viral genomes are small, so you can't have a lot of proteins dedicated to building a particle. You don't have enough genetic space. This is the theory of genetic economy. So they thought there must be a few proteins repeated over and over and over again to build either a round or a rod-like virus particle. So that was their contribution. And they developed that into a theory where they said, okay, for these rod-like viruses, the proteins are a few proteins repeated over and over again. They are distributed with helical symmetry. And for the round viruses, they are distributed with icosahedral symmetry. So I'm going to go through both of those with you and explain what they mean. But Watson and Crick made a big contribution to our understanding of structure, which of course was subsequently refined by other individuals as well. So there are rules of symmetry that we can use to understand these virus particles. They're very simple. First of all, rule 1, every subunit has identical bonding contacts with its neighbors. So basically you have the same protein or just a few repeated over and over again, and they interact in similar ways as a consequence because they're the same protein. And that gives you a symmetric arrangement, and it gives you a lot of stability, as we said before. And the second rule is these interactions are usually non-covalent, as I said earlier. To keep metastable particles metastable, you have to make the bonds non-covalent. And this non-covalent assembly not only gives you reversibility in the virus coming in, but it's also it gives you error-free assembly. So as we'll see later when we talk about assembly, if there's a mistake made in the cell during assembly of a particle, it can be reversed and corrected. And you couldn't do that, of course, if you had a covalent bond. Symmetry and self-assembly are beautiful things. Many capsids will self-assemble. And in fact, you can make the proteins in a cell and they will self-assemble into what we call virus-like particles. They are empty capsids. And we have a couple of vaccines that work that way. There's no virus genome present. There's just the gene encoding the capsid protein, which is just a part of the genome. And you can put that in a cell and have the proteins produced. The hepatitis B virus vaccines, the human papillomavirus vaccines, HPV, are virus-like particles made in yeast. So we take advantage of this symmetry and self-assembly properties of viral proteins to make vaccines. Here's how it works. First you have to know what viral protein is in the capsid, but you then can take it and you synthesize it in cell culture. And in the case of the papillomavirus, which is shown here, one subunit, the single-folded polypeptide chain, 5 of those assemble into the structural unit, which is a pentamer. We call it that because there are 5 subunits make it up, 1, 2, 3, 4, 5. And then those pentamers assemble to form the virus particle. And there you see it on the right. And these are colored. You don't see it very well on the screen here, but on your PDF, you could probably find the red subunit here in this one pentamer. So again, this is an empty particle. It is non-infectious. It is completely without danger. And it's really important. It prevents cancer, as we will see. So let's dissect helical symmetry. Here's an example of a virus built with helical symmetry, tobacco mosaic virus. The coat protein molecules. So this capsid is made up of individual coat proteins. They're shown at the lower left here in yellow. And they each join with each other to form this helix. And that is what makes the particle. Each of these little nubby things is a single coat protein joined with each other, and they wrap around to form a helix. And they're also binding to the RNA, which is inside of it. That's shown in the right-hand diagram there. So the coat proteins bind not only to each other, but to the RNA, and that forms the virus particle. That is helical symmetry. They're making a nice helix. And there's the electron micrograph of tobacco mosaic virus. That's it. It's a naked helical particle. There's no membrane. It's just that. And it's made of one protein repeated many times, wrapped around the RNA. So as you can imagine, the protein is protecting the RNA. And there are protein-protein contacts that are symmetrical and lead to the formation of this helix. So you can see the sizes here, about 18 nanometers in diameter. And that bar is 10 nanometers in length. Animal viruses are also built with helical symmetry, but they're always enveloped. Only the plant viruses have naked helices as the virus particle. So here's an example of a paramyxovirus, Sendai virus. It's a family of viruses, including measles virus. These are made with helical nucleocapsids. There it is right there with the RNA inside, and the single protein here, it's called NP, nucleoprotein, and that protein is repeated many times to form a helix, 1,000 nanometers long, and it's protecting the viral RNA. But in this case, the virus particle is then enveloped. This nucleocapsid is not naked as it is for TMV. So you can see here an electron micrograph of a measles virus particle with the nucleocapsid inside of it. This one's actually broken and the nucleocapsid is spilling out. So that's why we call it a nucleocapsid, because it's inside a membrane. In tobacco mosaic virus, it would just be a capsid because it's not a substructure. All right, so animal viruses with this kind of symmetry are always enveloped. A couple more examples of helical symmetry. Vesicular stomatitis virus is a rabies-like virus, bullet-shaped, very characteristic morphology, very popular as a vector. They use it now to make vaccines for other viruses. We'll talk about that at the end of the course. But it's got an envelope, and it has a helical nucleocapsid within it. It's RNA plus a protein called the nucleocapsid protein, or N protein, sorry, in this case. And on the right is the structure, the X-ray structure of the N protein. And you can see it binding RNA. There are 9 nucleotides of RNA there. So it's kind of a 2-lobed protein with a RNA binding cleft. Here, the protein is shown multiple copies of the protein binding a circular RNA. And in the filovirus particle, you make a very, very big helix. That's shown on the right here. The structure is solved by cryo-EM. And here is the RNA protein complex curling around and around and around. And it has a cap at one end. It's unusual. The other helical structures I've shown you, they're open at both ends. For some reason, this has a cap where it all comes to a point. And this whole thing is wrapped in an envelope, which are these 2 outer colors, blue and purple there. So another example of helical symmetry. And there are whole lots of viruses with helical symmetry. They're shown here. We'll revisit lots of those, but just so that you know, we've talked about measles, mumps, other paramyxoviruses like Sendai. They are envelope. They have a nucleocapsid, which is helical, as you can see in the picture. Rabies virus, we just talked about, a helical nucleocapsid with an envelope. Influenza virus is a virus with a helical nucleocapsid. It has it in 8 pieces in this case. Each RNA shown on the right here is wrapped in proteins, nucleoproteins NP, and then there are 8 of those. So that's the nucleoprotein. Filoviruses like Ebola viruses all the way here have helical nucleocapsids that are enveloped. You can see the curly helix there. So I think just by looking at these pictures, you can see the nucleocapsid is that nucleic acid protein assembly that is within the particle. It's the substructure. In these cases, it has an envelope around it. These, of course, are all RNA viruses. So here's the coronavirus. It happens to have a helical nucleocapsid, and there's the structure of it on the left. It has an envelope which contains glycoproteins, and inside is this long green RNA, the nucleoprotein plus RNA. That's the nucleocapsid. And in this case,
he said, inside of this is this green stuff is the nucleoprotein plus RNA, nucleocapsid, right here, inside. You got your lipid bilayer, the hemagglutinin, the spike protein, which we're very familiar with what the S protein is, the membrane protein, which is the little small things inside of it, and then you have the envelope glycoprotein, and then he talked about the green stuff. Now, to the right side of the black and white image over here, what you can see is the actual coronavirus live. That's the live virus. They have a, I know y'all heard Shepard say this before, but he said, man, he was impressed with science when he seen that they actually had a picture of the live virus. So there you go right there. There go that live virus right there. You can see it. It's, it's ugly looking. And that's why the example used is similar to that. So, again, I'm trying to prepare y'all for Monday when the virologist is on the Pseudo Killer Show, and we're learning as we go. And these are things that are, these things should pose questions, right? So let's move on.
It's 29,000 bases long. It's quite long. Now, when the first patients had this virus infection, the virus was isolated from them. A few times ago, I told you about cell culture experiments to identify this virus. At the same time, they took electron micrographs of it. And here's one that was published in the New England Journal. It's not very good, unfortunately. This one on the right is a better photograph, but it's not of the new coronavirus. But I want to show it to you because it shows you how this virus got the name coronavirus. When they first saw these particles in the electron microscope, they said, Oh, it looks like there's a corona around each one. You see those little spike? Those are the spike glycoproteins, which correspond to these molecules. Spike here. Those are those by negative staining. They say, oh, it looks like the corona of the sun, right? You look at the sun pictures, you see this thing. So they called them coronaviruses. So on the left is from a patient in Wuhan. You can't see the spikes very well, but they're there. And that's why they call it coronaviruses. So these are, again,
I just got a text message, man, and I'm doing this live. I shouldn't even engage or say anything to it because it's pretty much private, but I just got a text message that my auntie just got diagnosed with COVID. So I'm trying to figure out how she's doing. So far, she has lost her taste and smell, no fever, any other symptoms right now.
envelope viruses with helical nucleocapsids. Both DNA and RNA viruses have helical symmetry, and here's a selection of viruses. On the top left, there are viruses of archaea, double-stranded DNA viruses. Archaea is the third domain of life, right? Bacteria, archaea, eukaryotes. And these have their own viruses, not as well studied as everyone else, but they're there and they're quite remarkable. So this has a helical nucleocapsid and it's enveloped as well. Another double-stranded DNA virus of archaea, which looks different. It's naked and it has tail fibers to attach to the archaeal host. There are viruses of bacteria that have helical symmetry with single-stranded DNA. And then for RNA viruses, there are the rod-like viruses. The tobacco mosaic virus would be a rod-like virus. These are negative strand viruses. And then there are what we call flexuous plus strand RNA viruses of plants, also with helical symmetry, but they're not rigid like TMV. They are flexuous, as the name implies. So quite a variety of helical symmetries. If you want to build your own helical virus, you get these buckyballs, these little magnets. And there's one. If you come to office hours, my desk is full of these. And over the years, students have given me many different colors. So that's why we can make really long ones with different colors. And that's just exactly the way a helix is made, by putting subunits and wrapping them around. They're fun. They're really good. And you can still order these. Buckyball viruses. Next question is, which of the following describes virus symmetry and self-assembly? A, the bonding contacts are usually covalent. B, the bonding contacts of subunits are usually non-covalent. C, each subunit has different bonding contacts with its neighbors. D, self-assembly does not occur. E, none of the above. So let's see how we did. The answer is the bonding contacts are usually non-covalent, which most of you got. They're not usually covalent. A, that's wrong. And nobody got C or D. Good. So they are non-covalent, usually non-covalent. That gives them metastability and reversibility. All right, let's move on to round viruses, spherical viruses. The question here, how do you make a round virus from proteins that are irregularly shaped? So there's an electron micrograph of poliovirus. Looks roughly roundish, right? And on the right is one of the poliovirus proteins, VP1, which does not look round at all. So how do you get from what's on the right here to the left? And here are some clues that drove the solution of this problem. Number one, all of the round capsids have multiple numbers of precise multiples in terms of numbers of proteins. And multiples of 60 are very common, like 60, 180, 240, 960, the number of proteins that make up the virus particle. Second clue is that these spherical viruses come in many sizes, from small ones like polio to Mimi viruses and cafeteria Rowenbergensis virus. But capsid proteins are usually between 20 and 60 kilodaltons in size. So in other words, as the virus particles get bigger, the capsid proteins don't get bigger, but simply the number of proteins that make up the capsid increases in number. So Rotson and Crick looked at all of this And they concluded that these particles must be built with icosahedral symmetry. So what is icosahedral symmetry? So here on the right is an icosahedron. It is one of the Platonic solids, has 20 faces. Each face is an equilateral triangle. And that arrangement of equilateral triangles gives you what we call axes of symmetry. There are 5, 3, and 2-fold axes of symmetry. There are 12 each. So here on this particle, the green is the 5-fold axis. And it's called that simply because there are 5 subunits around it. There are 5 triangles around it. The 3-fold axis in red has 3 triangles around it. The 2-fold has 2. Very straightforward. On the bottom, the individual axes are shown in a different view. So this arrangement allows the formation of a closed shell, a very stable closed shell with the smallest number of protein subunits, 60. You can make a very small virus particle with just 60 copies of one protein. So Watson and Crick came to this because the fact that you make round particles with irregular proteins, and as the particles get bigger, the protein sizes stay the same. And they decided this was the way it was done. And this has pretty much stood up since then. There are some exceptions, but most of these symmetrical particles, if they're not rod-like, they're built with icosahedral symmetry. So let's start with the simplest one. It's diagrammed on the left there. It's made of 60 identical protein subunits. So here the protein, the polypeptide is the same as the structural unit. So sometimes there is one protein that equals the structural unit, or sometimes there are multiple ones, as you'll see in a moment. But here the protein subunit is the structural unit. And on the left is an example of such a particle. We call this a T=1 particle. I'm going to explain T numbers in a moment. And each of these commas is a single protein. And there's 60 commas on this particle. And you can see that there are 5-fold and 2-fold and 3-fold axes of symmetry made by the arrangement of these proteins. These proteins all interact in an identical fashion, either head-to-head or tail-to-tail. So if you look at this, the heads of the commas have the round part. You can see head-to-head interactions or tail-to-tail interactions. So here's a great example of identical protein-protein interactions in a particle, fulfilling the original idea of the rules of symmetry that we stated earlier. Now, it's important to remember that although the proteins are arranged with icosahedral symmetry, it doesn't actually look like an icosahedron. An icosahedron is this geometric particle, but virus particles don't look like that. They actually look spherical if you look at them in the electron micrographs, and that's because the proteins are arranged with the symmetry of an icosahedron, but they are not flat and triangular. Okay, it's very important to remember. So that's your simplest icosahedral capsid. There are plenty of examples of viruses that are built this way. One of them is a parvovirus. Remember the virus that can infect you or your cat or your dog. It causes 5th disease in humans, can kill your pets. That's a parvovirus. And this is a small capsid, 25 nanometers in diameter, just slightly smaller than poliovirus, made up of 60 copies of a single capsid protein. And it contains a single-stranded DNA genome. So here on the bottom left is the structure of the single capsid protein. And parts of it are colored red and blue, yellow, and green, because now when we assemble them into the virus particle on the right, you can see the contribution of different parts of that protein. So there are 60 of these single proteins comprising this capsid on the right. The triangle is the subunit and the structural unit, which is one protein. Remember these kinds of viruses, it's the same thing. And you get 5-fold axes of symmetry. Like here, you can see 1, 2, 3, 4, 5 of the blue parts, which are down here in the protein around the 5-fold axis. And also 3-fold and 2-fold axes of symmetry. So again, this is a T=1 particle. And you have one protein, which is the subunit, and the the structural unit, that's the red area there, and then the structural unit, 12 of those go in to make the virus particle. So these are the smallest particles that we know of, one protein repeated 60 times. Now, how do you make bigger particles? You add more subunits. You don't use bigger proteins. You add more subunits. And by doing so, now you get slightly different interactions among the proteins. And so here is a virus particle made up of 180 identical subunits. Remember, the first one was 60. Now we have 180. It's the same protein. And when you do this now, when you add more subunits, the particle gets bigger. And as a consequence, you have two different kinds of interactions among subunits. You have pentamers and hexamers. And that's very simple. Pentamers are units of 5, hexamers units of 6. And we can find them very easily here on this particle. Here's a pentamer shown by the green pentagon. And there are 1, 2, 3, 4, 5 orange subunits around it. And then the red triangle is a hexamer with 6 of the purple subunits around it. They're all the same protein. They're arranged differently in pentamers and hexamers. It's a way you have to do it in order to make a bigger particle. So because of this, the original rules of symmetry had to be modified when people learned there were bigger viruses made this way. The original rules said all the interactions among the protein subunits are identical. But now they can't be anymore because you have hexamers and pentamers. And so they look similar, but they're not identical. And that's called quasi-equivalent. They all engage in head-to-head, tail-to-tail, but there are differences so that you now have pentamers and hexamers. And you need that to make a bigger particle. Quasi-equivalence is just a bending of the original rule saying the proteins interact similarly, but not identically. That's how you get a bigger particle. So quasi-equivalence, again, when a capsid has more than 60 subunits, anything greater than 60, each protein has a quasi-equivalent position. The properties in different environments are similar but not identical. And you can remember that most easily by remembering that there are pentamers and hexamers in these particles. Now let's get back to the T number, which I mentioned a couple of times. The T number can be described mathematically, but I prefer it to describe it to you structurally. T stands for triangulation number. It is the number of facets in each of the faces of this particle. So let's go through some of these and explain it that way. Here we have on the top virus particles with T equals 1 triangulation numbers. The structural unit is, the subunit is the same as the structural unit. The protein polypeptide is the same as the structural unit. And that's a T equals 1 because you see this polypeptide forms one of these triangles, and that's the definition of a T equals 1. You could actually calculate the total number of subunits in a virus particle by multiplying the T number by 60. A T equals 3 particle. Now the subunit, the polypeptide, there are 3 of those making up the structural unit, 1, 2, 3. And you can see that outlined in red there. So 3 compared to 1, the triangulation number is 3. Very simple. And the total number of subunits are 180. You can keep going up. You can do T equals 4, where there are 4 proteins in the structural unit. Here there are 12 proteins.
I'm gonna pause it right there because I don't want to draw the video out too long, and we've been on 1 hour and 35 minutes. Anybody can pick up the rest of the actual lecture and tune into that. So what I'm gonna do is I'm gonna end this, and no, I'm not. I'm gonna take some questions and answers if anybody got any. If not, then you know what I'm saying, I'm gonna see what the fellas want to do, and then we might after show it on Coffee Pie side. But Hold on just a moment for me. I know I see Sutek and T'Challa in the building. So if they got something for the people, man, won't y'all go ahead and get into that real quick.
Nah, man, I'm gonna just say peace to the people. Appreciate y'all for coming through, man. A lot of other platforms deal with a lot of entertainment, but here it's a lot of education over entertainment. So, man, salute y'all. Y'all the real MVPs for sticking in here with us, man. Appreciate y'all. Make sure y'all, I know y'all got them stimulus checks, man. Come on with it. Come on with it. We got them clothes. We got that merch. We got them books. We got everything you need, man. Come shop with us. What up, T'Challa?
What up? What up? I mean, I ain't really got much, let the videos speak for themselves. Peace to everybody that's tuned in, of course. And, what I'm saying? We hope that y'all can take this information and apply it and actually make it through 2021 like you made it through 2020. It's a whole lot of dangerous information out here. So, we just trying to, if not dangerous information, what else? Correct information.
Trimmer said her sister or somebody, whoever contracted it again, literally had it 3 months ago. So there go your antibody test right there. Some people were claiming that some people develop antibodies for 8 months, 6 months. No, it's always been around that 3-month range. Like you'll develop the antibodies to fight against it for a little while, and then the antibodies go away. And now you're up for it. I don't know what Brother Delani meant by no sound. I'm pretty sure everybody heard everything that was going on, so it might be something on his end. But yeah, that's how the antibody stuff goes. That's how we know you're not gaining immunity against this virus. You're not gaining immunity. So I'm gonna leave it right there, man. And I'm gonna see what the brothers want to do, and I'm gonna end it like that. Appreciate everybody tuning in, like the show, share the show. Don't forget to subscribe. Make sure you subscribe to Kofi Pie Side YouTube channel, and you might see us live again over there.
Ladies and gentlemen, the books is in. The books are in. Yep. They're in. They're here.